Good morning to one and all present here. We'll be starting with the basic part of enzymes today. So in today's lecture, we're going to learn something that is associated with enzymes. Some basic part of enzymes we're going to take today, which is we have already learned all these things, but for a quick revision, we'll be going with the same part. Okay. So now enzymes are said to be uh, self-replicating molecules. Enzymes are said to be proteins in their nature, which are helpful for reactions inside the cell. So what are the enzymes? Enzymes are biological catalysts. So these are catalysts in their nature. So what are catalysts? Can anybody tell me? Okay, fine. I'll tell you. Catalysts are nothing but these are molecules which are helpful to speed up the reaction. They do take part in the chemical reactions, but they do not get affected by the chemical reactions. Correct. So these are said to be mediators of converting one biological molecule into second one. Now, the enzymes are said to be the central of a biochemical process because every biochemical process that has been carried out inside the cell will be helpful as biological catalysts or will be helpful for conversion of substrate into products. Now, this study of these enzymes is said to be very much important because all biochemical processes which happens inside the cell are being attached with this enzymes. So, therefore, enzymes are said to be an important biological molecules, and the study of this enzyme is called as the subject enzymology. Correct? Right. Moving on. Now, some definitions that are been associated with this. The definition includes what? So enzymes are biological catalysts. We have already studied this, which are said to be in living cells. Now these are enzymes are said to be proteins in their nature. We have already seen this part inside second year of BSc, where we have learned biological molecules like carbohydrates, proteins, and lipids. So these enzymes come inside the part of proteins. Now, what are these proteins are helpful for? Why enzymes are said to be helpful? Because these speed up the chemical reactions in a particular cell or in a living organism. Now, an enzymes acts on catalysts and it is helpful for conversion of these specific reactants into specific products. Therefore, these reactants are called as substrates. Correct? So these reactants are called as substrates. And once these substrates are there, the substrate gets converted into products. So therefore, a very simple equation is there. Whenever a substrate gets converted into product, who does it? The enzyme does it. Therefore, the equation becomes ES, that is enzyme plus substrate, is equal to enzyme plus product P. So enzyme remains as it is. It just converts that substrate into product P. Now, what are the chemical nature of this particular enzymes? Its chemical nature is said to be something wherein the enzymes are said to be proteins in their nature. One of the exception for this line is said to be ribozymes. Okay. It acts on the substrate and changes into the product. This is a very specific reaction that the enzyme takes place or the enzymes work on top of it, that it actually runs in this way. Then its molecular weight is ranges from few thousand million Daltons. It is very slow or very low amount of molecular weight is there. Therefore, it acts very specifically into the product. Then now, some enzyme proteins are modified covalently by phosphorylation, glycosylation, other processes. Various different types of modification can take place in this enzymes also. They are colloidal in nature and heat liable and water soluble. There are enzymes. Since enzymes, the structural enzymes are said to be proteins. The nature of the enzyme is said to be protein. Therefore, these are heat liable. Whenever a protein is being exposed to heat, the structure of that particular enzyme will be distorted. It is absolutely non-functional. They can be precipitated by protein precipitation reactions, amino sulfate and triglyceride. Means they can be removed from that particular systems with the help of these chemical reactions. Now, structurally, the functional enzyme is a hollow enzyme with proteins, apoprotein, and coenzyme. Now, see, if you see the hollow enzyme, inside the hollow enzymes, the hollow enzyme consists of all sort of things. If you see here, if you see my cursor, then this is the apo enzyme. This is the enzyme. This is the cofactor of that particular enzyme, and the substrate at which the substrate could bind. You can see the active site, and this is the coenzyme, which is helpful for binding of that particular substrate. Prosthetic group or coenzymes are metal ions that is very tightly or evenly covalently bonded to the enzyme. These are the coenzymes of the prosthetic group that is being present in here. Single peptide, polypeptide chain, oligomeric enzymes, lactate dehydrogenases, multi-enzyme complexes means these are categorizations of enzymes based on their abilities to degrade the substrate into product. The term cofactor, what are these cofactors? They are collectively termed to include coenzymes and cofactors. So coenzyme is an organic cofactor. They help in either maintaining or producing active structural conformation of enzymes. They help in formation of enzyme substrate complexes. Cofactors help in making structural changes in the structure molecules. Cofactors, what are these cofactors are helpful? These are helpful for binding of that particular substrate and converting the substrate into product. Cofactor accept or donate electrons. Now, how this chemical reaction takes place? By donation or acceptance of electrons. Formation of ternary complexes with enzymes or substrate. It means certain amount of mid-reaction would also take place because of cofactors. See this, you can see the cofactors, specific enzymes and their ions. All our cofactors are maximally of metal ions, basically copper, potassium, magnesium, iron, manganese, nickel, so on and so forth. And these are all specific enzymes that are been associated with it. Now, coenzymes. The non-protein inorganic lower molecular substances associated with enzymes are called as coenzymes. Coenzymes are metal ions that are very tightly or covalently bound. Coenzymes are second substrate, affiliated with the substrate, comparable with the substrate. Now, whenever a binding of molecule takes place, this binding will be helpful with some kind of interaction. So, what are these interactions? These are interactions that are associated with the coenzyme. Basically, the metals, the metal ions that are present inside the coenzyme will be helpful for uptake or giving of the particular product. Now, these participate in various type of reactions, oxidoreductant reactions, group transfer reaction, isomerization, covalent bond formation, so and so forth. You can see here the coenzymes have their enzyme structures that are associated with it. So that's all we have been able to finish up with this today's part. This was the basic of enzymes that we have encountered. So in the next lecture, we will be dealing with detailed structures of enzymes and certain enzyme classification as well. Okay. Thank you so much for your patient listening.